by a Y-shaped fork. Sap vacuoles absent. Instead, gas vacuoles occur in number of gases. Membrane-covered cell organelles, including endoplasmic reticulum, are absent. Rhizosomes are 70s in nature, bind to fission in the mode of multiplication. Varied nutrition including photoautotropic, saprotrophic, parasitic and chemotopotropic. Photoautotropic forms possess bacteriochlorophyll instead of typical chlorophyll. Flagella. If present a single stranded, they are made of protein and flagellin. Occurrence. Bacteria are the most abundant microorganisms. A handful of soil may contain hundreds and thousands of them. They are ubiquitous, being found in all places where organic matter is present. In water, air, soil, over and inside the bodies of various organisms, they can tolerate extreme environments like hot spring, frozen waters, desert, deep ocean, acidic, alkaline, and saltous conditions. Structure of a bacterial cell. It is a prokaryotic cell in which genetic material is not organized in the form of nucleus but instead lies freely in a necked supercoiled state in the cytoplasm, whereas it is known as prochromosome or nucleoid. Prokaryotic equal to eukaryotic cells are known for their rapid multiplication. The average size is 2.0 to 2.6 long and 1.1 to 1.5 wide. The smallest bacteria cells are 100 to 200 nanometer. The smallest bacterium is Dilister pneumostens. It has a size of 0.15. The largest bacterial cell belongs the spirosacids and blue-green algae. Here the cell may reach a size of 500. Epiolosicum Phycelsoni, a bacteria found in the intestine of brown sturgeon fish, and marine bacteria Thymarigenta remibinensis is. Therefore, some bacterial cells are quite large as compared to eukaryotic cell. In shape, bacterial cells are many types. Mycelia form is found in actinomycetes. Coccus bacteria are spherical or ovoid in outline depending upon their grouping. They are called monoecious, occurring singly. Diplococcus in twos, Tetrococcus in tetrads, Streptococcus in chains, Staphylococcus irregular shape like clusters, and Sarcina dimensional geometrical forms. Bacillus, the bacterium is straight and cylindrical like a rod, with ends being flat, rounded, or cigar shaped. It has three spatial types: Diplobacillus in twos, palisade bacillus like a stack, and streptobacillus in chains. Spirillum, the bacterium is coiled like a corkscrew. Spirillum, spirochat, aggregation does not occur. Vibrio. The body of the bacterium is like a comma, curved rod, or single turn of the spiral. Example: Vibrio cholerae, like Spirillum bacteria. The vibrio forms life singly. Stalked. The bacterium possesses a stalk. Example: Colobacter. Budding. The bacteria is swollen at places. Example: Rhodomicrobium. Flagellation. Depending upon the presence or absence of flagella, bacteria are grouped into flagella and non-flagellate types. The various forms of flagellation are as follows: a tricus, flagella absent; or monotricus, a single flagella occurs at or near one of bacterium; amphitricus, a flagellum at each of the two ends; lopotricus, a group or tuft of flagella is found only at one end; cephalotricus, a tuft or group of flagella occurs at each of the two ends or poles. Many authors use the term amphitricus for both single flagella and tuft of flagella at each end. Peritricus, a number of flagella are distributed all over the surface. Gram-positive and gram-negative bacteria. The grouping is based on the reaction of bacteria to gram stain, Kristen Gram, 1884. Bacteria stain first with weakly alkaline solution of crystal violet or Genetian violet. When all of them pick up blue color, they are then treated with 0.5% iodine solution, followed by washing with water and then absolute alcohol or acetone. Bacteria which retain blue or purple color are known as gram-positive bacteria, example Bacillus subtilis. Bacteria which do not return any stain and become colorless and termed as gram-negative bacteria, example Escherichia coli. Gram-negative bacteria are commonly stained with safranin. Washing of the stain in gram-negative bacteria is due to high lipid content of shell wall, which gets dissolved in organic solvents like acetone. Then difference between gram-positive bacteria and gram-negative bacteria. Gram-positive bacteria, they remain colored blue or purple with gram stain even after washing with absolute alcohol or acetone. The wall is single layered, outer membrane is absent. Thickness of the wall is 20 to 80 nanometer. The lipid content of the wall is quite low. The wall is straight. Murine or mucopeptide content is 70 to 80%. Basal body of the flagellum has two rings of swellings. Mesosomes are more prominent. The bacteria are more susceptible to antibiotics. Fewer pathogenic bacteria belong to gram-positive group. Porins are absent. Cell wall contains teichoic acids. The next is gram-negative bacteria. The bacteria do not retain the stain when washed with absolute alcohol. The wall is two-layered, outer membrane is absent. It is 8 to 12 nanometer. The lipid content of the wall is 20 to 30%.